The nuclear export receptor, CRM1, exports over 1,000 proteins from the nucleus to the cytoplasm. CRM1 binds nuclear export signals, or NES motifs, in cargo proteins. NESs are 12 to 15 amino acids long and contain 4 to 5 key hydrophobic residues. They are highly diverse in sequence and can bind to the hydrophobic NES binding groove of CRM1 in many conformations. A mutation of CRM1, E571K, occurs in several cancer types. The mutation initiates tumor formation in mouse model B cells and alters subcellular localization of around 50 proteins, but it is unclear how it drives tumor formation. Residue 571 is in the CRM1 groove near where NESs bind. We set out to understand how this mutation alters CRM1 NES binding and nuclear export. We tested 26 diverse NES peptides and found 3 that bind CRM1 close to residue 571. The mutation of the residue from glutamate to lysine causes CRM1 to bind less tightly to the NESs of EIF4E transporter and RPS2, but more tightly to the NES of MEK1. But do these differences in NES binding affinity affect the transport of these cargoes by CRM1? Using CRISPR, we generated HEC293 cells that carry this CRM1 mutation to see what happens to RPS2, MEK1, and EIF4E transporter. We found that the subcellular localization of RPS2 and MEK1 were unchanged. It turns out that the NES is buried in full-length RPS2 and partially buried in full-length MEK1, meaning that RPS2 is not a CRM1 cargo and MEK1 binds differently from its NES. However, EIF4E transporters showed decreased nuclear export in HEC293 cells and in patient cells with this mutation. Thus, our work begins the search for the CRM1 cargoes whose mislocalization in CRM1 mutant cells leads to cancer.